In our previous module, we saw how cells produced cyclic AMP in response to epinephrine. And we said that cyclic AMP is going to mediate cause and effect in the cell once the cell has bound epinephrine to its epinephrine receptor. Let's see what cyclic AMP can do. For example, in some cells, it can activate an enzyme called protein kinase A. It is also called cyclic AMP dependent protein kinase, or CAPK, or pKa, one and the same thing. The diverse effects of cyclic AMP are mediated through cyclic AMP dependent protein kinase. This is a kinase which can attach phosphate groups to specific serine and threonine residues in a protein. We know serine and threonine are amino acids. Once an amino acid has been incorporated in a protein, it's called a residue. Phosphorylation of many enzymes increases their catalytic activity, whereas phosphorylation of other enzymes inhibits their activity. We'll see examples of both these scenarios. First of all, let's see how cyclic AMP activates pKa, protein kinase A. Protein kinase A in the cytoplasm is inactive form if the cyclic AMP is not present. It is made up of 4 protein subunits, 2 catalytic subunits, 2 regulatory subunits. These regulatory subunits have 2 cyclic AMP binding sites each, 1 and 2. First of all, one cyclic AMP molecule will bind one site. It will cause a conformational change in this regulatory subunit of pKa, which will expose the second binding site for cyclic AMP. It is called the cooperative binding. And when that happens, the regulatory subunits, once they have bound 2 ATP cyclic AMP molecules, they will detach from the catalytic domains, catalytic subunits. Now the catalytic subunits of pKa are active. They can go out and do business. One of the things protein kinase A does in liver, for example, is once it is activated, it will phosphorylate glycogen phosphorylase kinase. It is also an enzyme, and as the name suggests, kinase, it attaches phosphate groups to other proteins. Once GPK, glycogen phosphorylase kinase, is activated, it phosphorylates and activates glycogen phosphorylase. Phosphorylates glycogen, which results in removal of one glucose molecule. Cyclic AMP simultaneously inhibits glycogen synthesis by, first of all, inactivating or suppressing phosphoprotein phosphatase, which is an enzyme which removes phosphate groups from other proteins. It is reverse of kinase. And pKa also phosphorylates glycogen synthase. Glycogen synthase in its phosphorylated form is inactive. Here are 2 examples of 2 enzymes which are activated by addition of a phosphate group. Glycogen synthase is, on the other hand, inactivated. And the net result is production of glucose. One glucose molecule is released by the liver cell, made available to the rest of the body. Once the pKa response is terminated, pKa has become inactive. Phosphoprotein phosphatase is no longer suppressed. It removes the phosphate groups from GPKs and GPs, glycogen phosphorylase kinase and glycogen phosphorylase. They both lose their phosphate groups. And it also removes the phosphate group from glycogen synthase, making glycogen synthase active. Now this glycogen synthase will start adding glucose molecules to the glycogen. So it is very important that cells are not doing things which are counterproductive. On one side, on one hand, it is degrading glycogen, and other system is making glycogen. So this is how the system works very beautifully in response to epinephrine in the environment. Here you can see the structures. Here, for example, we have glycogen. Glycogen synthase adds glucose molecules to the glycogen. Glycogen phosphorylase, on the other hand, adds a phosphate group, resulting in release of one glucose molecule from the glycogen. So this is the overall schematic which we just saw. The protein kinase A is activated in response to cyclic AMP, which is produced by adenyl cyclase, which was activated by G protein, the alpha subunit of the G protein, which dissociated from beta and gamma in response to epinephrine. When epinephrine attached itself to the receptor protein, epinephrine receptor on the liver cell, the cyclic AMP bound to the regulatory subunits of protein kinase A. The complex fell apart. The catalytic subunits were activated. These catalytic subunits then phosphorylated proteins, enzymes that resulted in release of glucose from the glycogen. So this is a beautiful system, an example of how G protein linked receptors work.